Thank you, Dr. Lin, for a nice introduction. And I also thank the Omics for having me chair this mini symposium. This symposium will be focused majorly on the molecular mechanisms involved in ocular tumors, in specific to retinoblastoma and melanoma, which Dr. Mary Beth will be talking after a short while. And we will be focused at what are the strategies and what are the targeted therapeutic interventions that are involved. I will be talking about the selective ablation of ocular tumor cells and in the presence of cold atmospheric plasma. So I come from this medical device company that works on plasma technology, which this is not the blood plasma. This is slightly an offshoot, and it is a new therapeutic intervention in terms of tumor cancer therapy. So what is plasma? This is the fourth state of matter. And so apart from solid, liquid, and gases, you have what is known as the atmospheric plasma. It could be in the form of helium gas, argon, or any other source of the plasma. In this case, right now at the company, we use either argon plasma or helium as a source of making the plasma gadgets or scalpels. We make both the scalpels that uses argon technology as well as the helium gas as a source. It not only, the scalpel is used for bloodless surgeries, and apart from that, the property of plasma is that it ablates the tumor cells. And how exactly it destroys the tumor cells selectively by not destroying the normal cells is something that I'm trying to study, and we will go through in a short while. This is a new technology in the field of cancer biology, and the world's first the cold plasma application on colon cancer patient was performed in the month of March in Baton Rouge. The application of ionized gas in the form of cold atmospheric plasma has been used as anti-tumor therapy, and Dr. Kedar, who is from the George Washington University, has been instrumental in developing this technology, who also collaborates with us in developing newer generation of products. How does this normally function? It's been reported in literature that the cold atmospheric plasma induces cell stress by means of redox potential or by modulation of the ROS. This is what is known so far. There's not much of literature. There is no mechanism, there is no clear definition to say that this is the pathway which is involved in inducing apoptosis in cancer cells. And my interest has been in the field of eye research because all my training has been from the Eye Institute or back from the Aravind Eye Hospitals. So my interest in retinoblastoma continues for a long time, and therefore I thought I should have the cold atmospheric plasma applications in ocular medicine as well. And therefore, apart from breast cancer, colon cancer, and other cancers, we also study the retinoblastoma in the lab. What are the pathways that are involved? ROS mediates cell stress. NF-kappa B has been associated. People have demonstrated that NF-kappa B is associated in the cold atmospheric plasma mediated cell stress. This is something that was there already in the literature. But what induces at the cell surface is something we wanted to know. And this generated the hypothesis for us to understand what are the receptors that are involved at the cell surface that trigger the downstream signaling targets. So in order to study that, we thought one of the important receptors at the cell surface is what is known as the death-inducing receptor 4 or trail receptor 1, which is a tumor necrosis factor, which is related to the apoptosis-inducing ligand. This is an important factor because people have shown in the literature that trail itself can be used as a chemotherapeutic drug for breast cancer patients or colon cancer patients, and this induces cell death dramatically. So is it possible that trail is involved in cold atmospheric plasma applications is something that we will see. This was the hypothesis that that I tried to generate at the lab. In a normal cell, the amount of trail receptor the cell surface is minimal. In a tumor cell, in a normal tumor cell, the receptor level is upregulated. But what happens when there is induction of cold plasma? There is increased ROS generation. So that elevates the amount of trail receptors at the cell surface. This induces the downstream signaling targets. It induces cell stress. The mitochondrial cell stress is increased. Sorry. And this induces various apoptotic signaling pathways to be activated. The BCL2, NF-kappa B. Is P53 involved directly? Does cold atmospheric plasma induce stress, cell stress at the DNA level? Does it induce a nick at the DNA? And does that create a loop and increase the trail receptor at the cell surface? We do not know as of now. All that we see is that there is an elevated expression of trail. So this is suggestive to say that trail can be used along with cold atmospheric plasma as a chemotherapeutic drug in cancer patients. The amount of trail that is given can be reduced, in fact. These were the methods that were used in the study. The cold plasma setup, which I will show in a while. We have the confocal microscopy. Most of our assays are using the trypan blue to study the dead cells and the live cells. Western blot and some of the other assays, which I will be discussing tomorrow in my talk. And apoptosis to detect the dead cells and some of the cell culture techniques. This is the equipment that is used. This is the equipment that was generated in-house and built in-house. On top is the cold plasma generator or the converter box. And this is the 601 generator, which is clubbed together, which is required for the conversion of the hot plasma to a cold plasma. Helium is used as the source, which is lying behind in this tray. This is the, in the laminar hood, you will see the thermal shaker, and there is a scalpel which is standing right there. The plasma is in the insert in B, you will see the setup with the cold plasma scalpel, where the plasma beam is used to subject the cells to cell stress and induce apoptosis. This is the handpiece or the scalpel. This is how it appears. And this is manufactured in-house and designed and manufactured in-house. What happens to these cells after cold plasma treatment? In this case, we tried to study the tumor stem cell. This was a generous gift from Dr. Brigitte Marianne from the University of Austria Medical Center. And this was a model for us to study the retinoblastoma and other cell lines in vitro. This cell line was generated in her lab from the patient, and this is very close to the tumor stem cells in the colon. A time-dependent study was carried out. This is a simple trypan blue dye exclusion test at 5 seconds and up to 3 minutes. H2O2 is a well-known indicator for inducing cell stress, and that was used as a positive control, and no treatment was used as another control. And helium alone is a control for the cold plasma. You would see here that there is a gradual increase in the cell death. After about 2 or 3 minutes, you would see that the cell death has gone up to 65% alone, and this is in the tumor stem cell. This is after 5 doses of treatment. But this does not happen in case of the other cell lines, where you would see that just after 1 treatment, there is enough cell death. These stem cells are more resistant, and you require more dosage of cold plasma. Helium does not show much of, there is no significant increase in the cell death. And after 6 treatments, the number of cell death increases, which is close to 85% of cell death. And the positive controls show similar pattern. We went ahead to study the colon stem cells which were isolated from the patient. This patient whose tumor was excised and the cold plasma was sprayed, the sample was examined for the presence of stem cells, and this is without cold plasma treatment, which was done ex vivo from the patient sample. And you would want to appreciate the amount of trail receptor at the cell surface. The green indicates that it is the trail receptor expression. Red indicates it's a CD44 marker positive for stem cells. The amount of expression for green is reduced when compared to the next slide here, where immediately after 1 minute of plasma treatment at about 1.4 watts, which is a very small voltage, there is an elevated expression in the tumor sample. This is from the patient sample. Although there is presence of stem cells, you do see that these cells co-express trail, and there was an elevated expression. So these samples were expanded in culture, and we wanted to see what happens after you ex vivo expand these cells which were treated with cold plasma and without cold plasma. So 2 different methods were utilized in this study. One is the explant culture, the other is the isolated cell culture. On the left is the explant cell culture, where there was no outgrowth of cells. But on the right, you would see that all these cells, which are rounded off, the arrow pointing towards, are all cells that are going through apoptosis. They are shrinking. They are no longer elongated, as you see in some of the other cells. This is after 17 days in culture. In the presence of cold plasma, there is induction of cell death and apoptosis, which is also seen after 20 to 27 days in culture. However, when the liver samples which were not treated with cold plasma, the colonies grew in size, they were healthy and proliferating even after 20 to 27 days in culture. This is from another area where the sample was excised, and you see that the cultures on the right show cells that are clustered and they were floating, and these were all indicative of dead cells. On the left are explant cultures. There were a couple of few fibroblast-like cells, and this was from the subphrenic diaphragm, and we think that this was from the normal, healthy remnant portion of the tissue, and this was not of colon cancer origin. And in this slide, you would appreciate that the cultures that were generated without the cold plasma treatment, the colony size increase. This is typical of a stem cell culture, where the colony size increases. The colonies were proliferating in culture, and after 27 days, you see that the patient still had the potential to develop into tumor even after the 4th stage. So this patient was referred to Dr. Kennedy, who is the principal investigator for the cold plasma program. And she was in the 4th stage of her cancer. And after treatment with cold plasma, this patient is healthy and free from chemotherapeutic drugs as well. So therefore, this technology was further extended. I will probably go into the next slides, where I will be discussing more on the retinoblastoma cells. This slide shows that we used a couple of markers, like the CD44 and Ki67 in green, to show that without cold plasma treatment, these cells were still proliferating in culture and dividing. Whereas in the presence of cold plasma, the nucleus was disintegrating. There was fragmentation of the cells. There were just very few remnant cells that were alive, And this is a slide that shows that without cold plasma, there were almost 24% of cells that were double positive for the proliferative marker as well as for the stem cell marker. However, this population was missing in the presence of cold plasma. And most of the cells that were remaining were all disintegrating after 1 month in culture. The same technology was extended to retinoblastoma. And this was an extensive study that was done using the ARPE cells, which are used as a control cells, and Y79 retinoblastoma cells were used in this particular study. And a time course assay was carried out, and we see that the percentage of cell death in the presence of cold plasma after 1 minute and 2 minutes was minimal, bare minimal in the ARPE control cells, as opposed to Y79, which showed 87 to 90% or more cell death. So this is what we consider as a threshold, and this was used, these two parameters of all 1 minute and 2 minutes were considered appropriate for treating the retinoblastoma cells, and all our future studies were carried out with these two parameters alone. This is a slide to show the surface expression of trail receptor. Trail receptor is a TNF-associated receptor molecule which induces cell death, which is also known as the DR4 receptor. And on the top row is the tumor cells, the Y79 retinoblastoma cells. You would see that in the helium-treated, which is the control for the cold plasma, there is no or minimal expression of trail receptor, but the green surface expression goes up. These are confocal, three-dimensionally merged images which were quantified, and you would see the quantified number in this particular lane. There is about 8,000 or so mean pixel intensity. The total fluorescence intensity was quantified. It was significantly more in the tumor cells than the control cells, control ARP cells. Even at 2 minutes, the same kind of pattern was observed. The second row shows the cells that are labeled in red, which indicates that it's a marker for cell stress. Cell stress was observed only in the tumor cells in the presence of cold plasma. Such cells were not observed in the ARP, even in the presence of plasma. So there is a differential effect of cold plasma in the retinoblastoma cells and the normal cells. H2O2 was used as a positive control, and most of the cells were positive for cell stress inducer, which is a mitosox, which labels the enzyme dismutase. So therefore, it gets oxidized and appears red in the mitochondria of the cells. The lower panel indicates the ARP cells, which do not show or very bare minimal expression of green trail receptor expression at the cell surface. There is no mitosox ROS positivity in the panel below. So in summary, we say that cold atmospheric plasma induces cell death of most of the primary colon cancer stem cells. Tumor stem cells in vitro requires more dosage of cold plasma. A single dose was not sufficient, but however, we have shown in the patient who was treated, there was 1 dose of plasma treatment was sufficient enough to destroy the tumor cells. So therefore, the in vivo and in vitro cannot be compared, and it is a total different system. The same technology was extended to retinoblastoma, and I would be showing some of my other results from the molecular biology as well as the microscopy results tomorrow. And retinoblastoma responded to cold plasma and induces cell death at about 1 or 2 minutes at 1.4 watts. There is elevated expression of the death receptor inducing molecule in the retinoblastoma cells. So these studies indicate that cold atmospheric plasma can serve as an alternative therapy in solid tumors. Most of the study was carried out by Ms. Elizabeth Williams, who is the senior research assistant. Dr. Warren Rowe does most of the Western blot studies, and Devin Saunders, who was involved in the molecular biology work. And this is the team. Thank you for your attention. So we'll probably have the discussion at the end of the symposium.